RNA molecules are made of single strands of nucleotides. RNA strands fold in on themselves, forming hydrogen bonds between nitrogenous bases and between bases in surrounding water. These bonds cause RNA molecules to form different shapes. Different sequences of bases equal different shapes. Hydrogen bonds form between bases in a predictable pattern. A nucleotide with an adenine base, A, will hydrogen bond with a nucleotide with a uracil base, U, A and U. A nucleotide with a guanine base, G, bonds with a nucleotide with a cytosine base, C, C and G. Originally, RNA played many roles that have now been taken over by more specific molecules. RNA's critical function that remains is the creation of proteins. Enzymes now catalyze reactions instead of RNA. Mitochondria store energy, and DNA stores genetic information. Its double helix makes DNA a better archive for genetic information, since the bases are protected inside of the helix. Also, thymine is more stable than uracil, but its helical structure means that DNA can't directly work in the cell. It is a library of information, but the only way that information can be used is via RNA. RNA is chemically active in the cell. DNA is not. DNA is useful and stable as an archive. RNA is useful working in the cell. DNA is maintained in a safe environment to maintain the integrity of the genetic code. RNA is used throughout the cell to implement the genetic code that is stored within DNA.